Hi, Whenzhong. Thank you very much for coming on to give an interview or give an update, sorry, I should say, for the MECFS community here through this through OMF research that you're doing. I thought we could cover maybe a number of topics. You're the big data analysis expert that we have, and I thought we'd start with kind of my first impression of you in 2017, I think, for the Stanford Symposium, when I saw you speaking about the severely ill patient study and the large amounts of data that you were handling. It became clear that you were dealing with massive amounts of data and very new technologies in the way of analyzing statistics as it pertains to big data. And so coming on from that, I guess I thought it was very interesting, the approach in terms of trying to generate as much information as you could for MECFS and to develop more understanding and information about the disease. I thought maybe we could start talking about maybe newer projects that you've been working on in terms of the data analysis component. One in particular I thought of great interest was some of this work with generating data from David Systrom's samples. And so maybe I thought you could maybe describe what they are and what that is, and what type of data you've generated, and where the analysis is up to on that.
Yeah, thank you. So I think the invasive CPAT study that you are referring to is one of those studies I think would potentially generate important insight to this mechanism in MECFS. The reason, as many of us know, is that MECFS is a complex disease involving a lot of organ systems. And using this invasive cardiopulmonary testing, we can simultaneously identify the dysfunctions in the physiology of the patients during exercise, as well as getting the molecular signatures from the circulating blood of the patients during and after the exercise test. In particular, we're looking at both the pulmonary blood as well as the arterial blood, which other technologies currently cannot easily have access to this type of information. So far, Dr. Systrom's lab has done hundreds of MECFS patients, as well as I think at least a couple thousand of other patients, for example, patients of heart failure or patients with pulmonary diseases. So that provides a range of diseases that we can learn from and compare and contrast with the MECFS patients. So far, we have done the cytokine analysis of the pulmonary and artery blood of the MECFS patients before, at the peak, and after exercise. And from there, the most striking finding that I think we found is that there's a significant increase in the arterial blood, those pro-inflammatory cytokines, comparing to the venous blood, which again, using other technology, it would be hard to get this information in those patients. And in particular, this increase is actually in response to the exercise itself. So we're still trying to figure out exactly which tissue, because when we think about the arterial blood versus the venous blood, the only organs in between are the lung and the arterial blood vessels. So we're trying to figure out exactly where these cytokines were generated. And that potentially can provide us new insight on how we might be able to potentially control this cytokine inflammatory response, and therefore controlling potentially post-exertion malaise. So that's one, I guess, exciting results that that we have gotten so far. And most recently, we have gotten the metabolomics data and the proteomics data from also the arteria as well as the venous blood of these patients. And our preliminary analysis shows hundreds of metabolites seem to differentiate between the patients and the controls at the peak and after exercise. And as we know, during exercise, the majority of the blood would go through skeletal muscle in response to exercise. So this might reflect something that's changing in the muscle of the patients comparing to the controls. So hopefully we can have more to report next time.
Yeah, that's really fascinating. I think obviously you spoke on the power of sampling within that that study with the invasive cardiopulmonary exercise testing, where they're effectively riding a bike, I think, and they have catheters collecting blood samples from the vein just before the heart, and then I guess the arteries just after the lung. So that's a kind of very unique type of sample that you're able to capture, and I guess looking at the difference before and after the lung and the heart, I think was kind of fascinating that you were able to see a different signature in ME/CFS patients, maybe indicating there's something creating cytokines somewhere along there. Pretty very interesting. I guess it's also, for other analysis that you're doing at the moment in terms of research, probably similarly to what you're working on here in terms of looking for cytokines, metabolites, some protein markers as well, would be some of this COVID to ME study, this collaborative research study that's happening, looking into the clinical trajectories of COVID patients. And as they get to what makes people go on to develop myalgic encephalomyelitis instead of recovering as they normally would. And that's kind of, as I can tell at the moment, some of that data is starting to trickle through. Where are we at with that research project?
Yeah, that's a terrific question. So I think the study is going very well as a result of this multi-center collaboration between the Harvard Center and the center in Uppsala in particular. And we're receiving samples and data from Jonas Bergquist's group on the COVID patients, as well as patients with related diseases. As you probably stated earlier, COVID provides a unique opportunity for us to actually understand the disease process for ME/CFS, because as we all know, a substantial percentage of post-COVID patients, unfortunately, would have a high chance of getting into ME/CFS. But on the other hand, in this case, we know that the etiology and the timing of the infection, which trigger this disease process. So that sort of provide us a unique opportunity to study the development of ME/CFS. In this particular study, we would also have access to the CSF sample, the cerebral spinal fluid sample, which is a unique tissue type for us to study. So right now, we looked at the proteomics of patients versus controls, and you can clearly see this neural information in those patients in the acute phase. The question obviously is, when the patient eventually, if the patient eventually develop ME/CFS, whether at that point we would still see a clear, neural inflammation in those patients. That remains to be seen, but at least in the first 2 months, we see a clear difference in the proteins in the CSF between patients and controls, which is a exciting finding, and hopefully this can help us to identify the disease mechanism and eventually treatments in these patients.
Yeah, I mean that's kind of that's the holy grail. That's what we're really after at the end of the day, and also just looking for predictive markers and some maybe understanding that etiology. I think would be fantastic.
Absolutely.
To go in. So I think also, I mean, I guess on that note of treatments, I think one of the more interesting sort of work that I've seen you actually been working on has been this look at the network analysis for treatments. And I guess this is kind of trying to identify. It's a complicated concept, but effectively what you're doing through data analysis is trying to identify treatments for ME/CFS, potentially repurposing of those. Could you maybe go over a kind of a description of that and some of the interesting avenues maybe that could potentially go down from your findings?
Sure. So by the way, that's obviously also a collaboration with you and your center. I guess the overall idea is to essentially compare the data that we have in ME/CFS with the data that people have already, or scientists have already collected in other diseases that are better studied than ME/CFS, for example, heart failure or neurological diseases. And as we all know, because ME/CFS is such an understudied disease, the data that we have is largely, scattered. So there's a need to actually integrate data from multiple studies. Each individual one of those studies seem to be of a small sample size and might only look at one aspect of the disease because obviously funding issues. And then compare this pattern with the patterns that are in other disease studies people have already established, and see which part of the signature in ME/CFS is similar to the part that people already know in other diseases, to see essentially whether there is a subset of well-known diseases that look similar to ME/CFS, and if so, whether there are proven drugs that are effective in those diseases that we might be able to consider in ME/CFS. So that's our current focus. And a step further would be to look at actually the combinations of symptoms in subsets of ME/CFS patients. For example, if the patient is mostly affected by, let's say, sensory disturbance, would there be other diseases that would also show similar combination of symptoms? and whether some of the drugs that are used in those diseases can be, as you mentioned, repurposed or tested in this subset of patients. So that's another area that we're currently looking at.
Yeah, I think these are very, very smart way. I mean, it's, as you describe it, to be quite, in a simple way like that. It's very, very different when you see it on a page and the complexity of the information that you're kind of creating here, and the pathways. I'm sure it'd be great, obviously, to see that in a presentation sometime soon, in PowerPoints and all that sort of stuff. But I think people get the picture in terms of how it's, the value of what you're doing. I think getting that across is really fantastic. I think that work is very important. In terms of, I guess, other research that you've been kind of conducting with ME/CFS, beyond the research, one of the key aspects is kind of this maintenance and the creation, I guess, creating the creation and the maintenance of the data center that we all utilize to kind of put data up and share with each other, but also able to kind of get insight from other people outside of us in the broader community, and also get other people within the science community to kind of upload their data if when possible. I think that's a really important resource for us working together on this disease. And then also, obviously, you've started developing the REDCap component to that, which is to try and integrate some clinical data. So I just thought maybe you could speak a little bit about that as well.
Sure. So on the data center side, as we just discussed, there's, I think it's essential for ME/CFS research, just because the current studies are limited in sample size, as well as the dimension of analysis, because again of this lack of funding. So each study can only typically look at a few patients and look at only one aspect of the biology. So there's this essential need to actually combine data from multiple studies in order to get a better picture of the disease. And this data center is established to facilitate this kind of collaboration. So right now, as you know, data from multiple OMF-funded data centers, research centers, and OMF-funded projects would put the data into the data center before the study is published. So that enable researchers in the entire community to have access to the research data and freely exchange their thoughts on the data and the findings before the publications. And I think that's, that's essential for research in ME/CFS, getting new insight in this disease. And the second aspect of that is, since we're trying to combine what we know in other diseases together with what we know in ME/CFS, getting this data as well as the interactions with other diseases also in the data center would facilitate us to generate hypotheses and test those hypotheses quickly, and as you just mentioned, considering the candidates for clinical trials. And the REDCap project is established to support basically this clinical study and try to enable the recruitment of patients using a uniform standard of the inclusion and exclusion criteria. And as we know that getting consistent patient recruitment, especially for complex disease as ME/CFS, is critical for the results and data to be compared across different studies. So that's one of our focus for this REDCap project. The other focus of this is to enable generating surveys across multiple research centers, and so we can actually survey patients and get information and pull information together.
Yeah, I think that's a fantastic resource and very, very instrumental to the work that happens around all the research centers, I think, in OMF to some capacity. Wayne, I think you're doing fantastic work, and thank you very much for updating the community on that work that you've been doing. Cheers.
Thank you.
No worries.
Thanks.